Hey everybody, I'm John Sterling. I'm the editor-in-chief of Genetic Engineering and Biotech News. Thank you for coming. There's a lot going on here this morning, so we're going to give it another 2 or 3 minutes for more people to come in. I even had trouble finding the thing this morning. Before we start, just a couple of things. We really want you to get involved in the discussion. Basically, it's a panel discussion where we're going to ask some questions. They're going to answer them. They're going to talk among themselves. I'd like them to bounce each other's ideas off each other, but we want you to be part of this. So 2 things. One, over on the table right near that pillar, there's some white cards and pens. If you have any questions, well, first question, if you want to just raise your hand. Anytime during the session, that's fine, because I'll be looking at you, and we'll take your question, because something might really move you, and we don't want you to forget it, so ask us right away. And there's also a microphone right next to the pens if you want to ask the question. If anybody wants to Twitter a question, don't feel like getting up or you just like using Twitter, my handle is @JohnSterling4. And I'll take a look from time to time to see. But we really want you to be involved. You have 4 really professional, experienced people in regenerative medicine here, in stem cells, in tissue engineering. It's a really great panel, so please, any question at all, do ask. Okay, so the format's going to be, I'm going to introduce each panelist individually. When I'm done, I'm going to ask each panelist to maybe take a few minutes to talk a bit more about their specific work and what they're doing, and maybe where they see that going. And then we're going to throw out some questions. It worked very well last year. What we want this year is we want your input. Again, you have access to 4 really experienced, a lot of expertise, so use this to your advantage. Okay, so 4 panelists. On the left is Dr. Aubrey de Grey. He's a biomedical gerontologist based in Cambridge, UK. And is it recently in Mountain View, California?
Well, for the past 5 years.
You got pretty views out there, right? More than mountains.
Mainly mountains.
Okay, good. And he serves as Chief Science Officer of the SENS Research Foundation. It's a California-based 501c3 charity dedicated to combating the aging process. He is also Editor-in-Chief of Rejuvenation Research. Dr. Jeanne Lai is Professor of Developmental Neurobiology and Director of the Center for Regenerative Medicine in the Department of Chemical Physiology at the Scripps Research Institute in California. Very, very fascinating place. I visited there some years ago. Dr. Loring and her team focus on human pluripotent stem cells, which can become every cell type in the human body. And Dr. Loring is a member of the editorial board of the journal Bioresearch and Open Access. I hope she also tells you that she's involved in doing some work with saving endangered species. She was just in Vienna, it was last week, or 2 weeks ago, where some people got together. I actually read a story about a white rhino, northern white rhino, died at 43, and there's only a few left. So, yeah, I'm encouraging you to say something about that. I mean, we're here for medical, but let's save the species too. Dr. Graham Parker, right there, blue shirt. He's an assistant professor of research in the Department of Pediatrics at Wayne State University School of Medicine. His major interests include the therapeutic potential and vulnerability of human stem cells and research on developmental models of neural disorders. He is editor-in-chief of Stem Cells and Development. And Dr. Peter Johnson is a principal of MedSurg PI, a medical affairs services company. He formerly practiced reconstructive surgery for 10 years at the University of Pittsburgh, where he founded and was the first president of the very famous Pittsburgh Tissue Engineering Initiative. It was one of the major tissue engineering efforts in this country, and we wrote about it when it happened and since, so we were delighted to have him. He subsequently co-founded and ran a number of life science companies, and he's co-editor-in-chief of the 3-part journal, Tissue Engineering. So as I say, you've got incredible experience and expertise here. Please, 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 in any moment, send up a card, use the microphone, send me a tweet. We want you to be part of this, okay? So I just want each person to talk, a few minutes, a little bit about their work. They're the stars, not me. I just wanted to give a brief introduction. Let them tell you what they do. Peter, why don't we start?
Hello, everybody.
Thank you very much for coming.
Hello? Yeah.
Great. Hello, everyone. Thank you very much for coming. I've spent my career trying to understand clinical medicine, particularly on the surgical side, and then understanding how technology can be cloned to serve many more people than we can with our hands alone. We didn't go back through my occupational history, but if you did, you'd see a fairly rational, stepwise approach that led me to the point where today I'm partner in a company that can serve all companies as virtual chief medical officer to help drive the technology forward along lines that we've heard at this conference.
Thank you. I'm Jeanne Lai. I'm currently a professor at the Scripps Research Institute. And my research and my passion revolves all about stem cells, epigenetics, and genomics. That's a really nice sort of group of technologies that I think has a huge potential. As was said, I am also, since I work on stem cells, sometimes those stem cells are human in most cases. I'm actively working on a stem cell therapy for Parkinson's disease. My lab is working on a stem cell based therapy for multiple sclerosis, and we're trying to understand the molecular basis of autism. In addition to that, the San Diego Zoo and my lab collaborated a few years ago, started a collaboration which continues today, to try to use stem cell technologies to rescue endangered species. So just last week in Vienna, there was the first meeting of a diverse group of experts from stem cell biologists to people who actually anesthetize zoo animals. And we all in that room had the expertise to go from a stem cell to a northern white rhino. We chose that species because there are only 3 living and none of them are reproducing.
I can follow anything. I can take a challenge. I'm Graham Parker. As mentioned, I'm the editor-in-chief of Stem Cells Development. I'm also the executive editor for Nucleic Acid Therapeutics, and that kind of describes my research interests. I got into stem cells once I decided that we weren't going to persuade people to stop smoking, so we should start looking at what the effects of nicotine are to the unborn child, the person that's not making the decision. And over the years, I've looked at a wide range of stem cell populations. My last funded research in neurology was making models of motor neuron disorders using mouse embryonic stem cells. And to try and sell you on the integrity of my approach to the regenerative medicine field, I will tell you how delighted I was at the OTS meeting to present the Young Investigator Award to a man whose research has made my research completely irrelevant, because the nucleic acid therapeutics advances that have happened to actually treat children with spinal muscular atrophy are incredible, and it basically has made what I was doing irrelevant. So the research I'm doing now is funded by the NIEHS, the Center for Urban Research on Environmental Stresses, and I hope we get to discuss how important it is that we start to become more collaborative with the public in our research goals. This is going to become more and more important.
I always thought you were a singer. There you go. So as you've heard, I edit Rejuvenation Research, another of the Liebert journals. Of course, I want to thank, as we all do, Marianne and Liebert for sponsoring this session. And I am here, really, as I have been almost every year that this conference has occurred, because the focus of SENS Research Foundation is specifically to attack the ill health of old age with regenerative medicine of one kind or another. Really rather broadly defined regenerative medicine. We do things not just at the level of stem cells and tissue engineering, but also what you might call molecular regenerative medicine, repair of damage within cells and even the extracellular matrix and such like. So quite a diverse range of things. We're a pretty small foundation based in Mountain View, California. We only have a budget of about $5 million a year, but we think we spend it quite well. And actually, I should probably mention that in this same room immediately after this session, with I think a 10-minute break for you to go and grab some lunch and come back, there will be a session that I'm chairing which is about the application of regenerative medicine to the problems of aging. One thing I should actually mention is that in addition to the excellent researchers who are listed in the program, is Amin Zhang from the Buck Institute and John Cook from Houston Methodist, we will also have one of my absolute superheroes, Mike West, taking part in the session. He's actually speaking right now in the Regency upstairs, but he's going to come down here and give... but he's going to come down here and give an abbreviated version of the same presentation, so you don't have to leave right now. So I very much hope you'll stay. As I say, you should just grab some lunch outside and then come straight back in. What I'm really here for is because Bernie Siegel and indeed a number of the other long-standing participants in this conference have been great friends of and visionaries understanding the concept that the most practical approach to combating the diseases of old age may actually be to turn back the clock, to genuinely repair the damage of aging, rather than to slow down the clock, as gerontologists have traditionally preferred to think about. And so I look forward to talking about that a little bit during this session, and also, of course, in the next session. Thank you.
I also want to give him a little plug. He wrote a book a few years ago. He's got some very interesting, and he's testing these concepts of aging. What's the name of the book?
It's called Ending Aging.
Okay, all right, and that's a pretty recent book, right?
6, 7 years.
Okay, great. We've aged in the meanwhile, so I hope nothing has changed. As you can see, they're not only a bright, interesting, expert panel. I worked with most of this panel last year, and they got a great sense of humor, and they're very relaxed. And believe me, I've been doing this for 30 years, and there were some panel discussions where it was literally like pulling teeth, but not with these guys. This is fine. So, as editor of Jeanne, so we developed questions that we thought would be really try to get what's going on right now and what's coming down the line, not so much looking at the past. But at editor of Jeanne, a lot of these questions are what we ask when we do a story on regenerative medicine or stem cells. And we always start out, and we're doing this right now at the beginning of the new year. What development has surprised you the most in regenerative medicine the past year? So over the last year, as we go into 2016, why don't we start down with Aubrey and then work our way across?
Oh, I had a really bad thing, hasn't
We're going to need that.
Well, I don't know about that. Graham suggests rather controversially that it's better to be first with hard questions like this than last, but I'm not sure. I like time to think. I guess the most surprising thing from my perspective as a gerontologist has been the rapidity with which, after a long period of very entrenched skepticism, the idea of repairing damage has suddenly become mainstream as a way of combating aging. I suppose I have to be, if I want to be strict about the timeframes, I have to say that this really became apparent perhaps 2 years ago, but it really started to gain momentum in the past 12 months, I think. When the Mayo Clinic published a paper maybe 3 years ago that emphasized the beneficial effects on phenotypes of aging arising from the elimination of what are called senescent cells, cells that get into a toxic state but then are resistant to pro-apoptotic signals, and so they hang around and accumulate. Yeah, that's one of the major themes of the work that I've been advocating for some time, and when it was actually done in mice by people who had plenty of credentials and reputation and everything, it seemed to really kickstart a whole new way of thinking within the gerontology community in terms of its interface with the regenerative medicine community. And so now I think we're seeing a lot more attention being paid to the idea of both traditional regenerative medicine in the sense of stem cells, and also more, left field versions of aspects of regenerative medicine by people who are interested in aging. Much more crosstalk. Institutes that are nominally focused exclusively on aging are doing a lot more regenerative medicine, and vice versa. So I would say, for example, the Buck Institute doing a lot of regenerative medicine, the Wake Forest Institute of Regenerative Medicine doing a lot more on aging. It's a fantastic thing to see, and I think it is. It's not a surprise in the sense of being poorly motivated. It's extremely well motivated, but it's a big surprise in terms of how much contrast there is in terms of the rate of take-up of these ideas relative to how things have been for a decade previously, where we've been trying without very much success to promote the concept.
Okay, so those of you that have suffered through my presentations before will know that one of my favorite hobby horses is characterization, standardization of technique. And so the thing that surprises me the most this year is how much venture capital has come back into cellular medicine and how much more public interest there is, where to my pea-sized brain, we really haven't got any further in actually solving those problems of standardization and characterization. So in a sense, it's a good thing, and I'm happy that money's coming back into the cellular field, but at the same time, I'm a little bit surprised.
Why do you think that is?
What, the money's come back,
Yeah.
or that we haven't gone on and done the characterization we have to do?
Why is the money coming in?
You asked me, what am I most surprised by? I want somebody to tell me the answer, because if I had any money to invest, I would be investing it in the nucleic acid therapeutics. I wouldn't be investing it in the cellular therapies.
Anybody have any experience in this? Might have an idea why money's suddenly flowing in, whereas they're not moving as forward as fast? Again, for those of you who came in late, we really want you to be extension of this panel. I don't want this to be a lecture. We've got some cards over there. You can ask a question, a microphone. You can also Twitter me at John Sterling 4. And at any time during this, we used to wait for questions at the end, but if something really resonates with you, please get up, say something, whatever, because, share it with the rest of us.
I'm Camillo Ricordi, University of Miami. Actually, I was equally surprised about the move back from even big pharma, not just venture capital, into cellular therapy, because up to a few years ago, big pharma would say if you can do it with a molecule, don't do it with a cell. And now there is a big shift from Novartis, Astellas, Celgene, and some of the major players. But the challenge, as you say, is standardization, especially in cell product. We just finished the phase 3 trial, FDA, for islet transplantation. It took 7 years to standardize manufacturing across 8 manufacturing facilities, with still the deep awareness that we have not at all a standardized product, because you start from different donors, different cause of death, different reagents. So it's very difficult to fit cellular therapy within a regulatory framework that is designed for new molecular entity where you can control the whole system. So I think that the challenge will be to develop a new paradigm with regulatory agency to find where cellular therapy can fit in a new regulatory framework. Uphill battle.
Actually, I would argue that the current state is good. Allow every party to do what makes sense for them, and let the evolutionary process of product development find out what works best. If we standardize everything right now, I don't know that we will standardize it to the best possibility. And the moment that something, that one approach or two approaches will prove good, will prove as a breakthrough, I would argue that the rest of the field would coalesce towards that area. I don't see a problem with currently, as long as you're safe, as long as you're up to regulations, do what makes sense.
Yeah, okay, so your point is totally valid, but when I'm talking about standardization of method, I'm not seeking to restrict what people are doing. What I want to make sure is that when you do something on your bench, it's exactly the same as what I'm doing on our bench, and we have a shared understanding of what that is that we've actually done. At the moment, we can't even decide what to call the cells themselves, let alone the technique.
That's up to actually the editors when papers are being submitted to say, okay, tell me exactly what you did,
Right.
such as it can be reproduced on the other side of the globe.
Go on, Jeanne.
Okay, so I get two because I also get to comment on his. So I was really impressed to learn this here that NIST, which is the National Institute of Standards and Technology, is the same guys that do the atomic clock and have the real kilogram, are now working on standards for human pluripotent stem cells. So they're coming up with some unusual methods, but their main objective is that you can be absolutely certain that you have the cells that you want. But what I'm going to suggest to Graham is that he put his foot down and require that any cell line that is used in his, that is published in his journal, have at least some kind of SNP genotyping or STR to know that it's a cell line that you said it was, and also that maybe the genome is intact. I mean, those are kind of important things. And there's an incredible amount of money wasted and heartbreak because people simply have the wrong cell line. I've been reading some articles about it now, and it's estimated up to like 80% of the cell lines published are not what people thought they were. It's not just HeLa either, yeah. Yeah, I won't let HeLa even on the floor or in the building. Okay, so let me tell you what I'm most surprised by. What completely took me by surprise was CAR T therapy, the development of immunotherapy that would be directed against your own immune cells, and as a last-ditch effort to clear up some forms of leukemia. I think it's I had no idea that this was going on. And it's so revolutionary that it's caught the attention of even pharma companies, which I think is, in a very short period of time, is quite a remarkable feat. And I think one of the reasons it interests me so much is that I think because these are mostly autologous therapies, you take the T cells out of a person, you modify them so that they will attack their own B cells, put them back in, they kill off all the B cells. The B cell leukemias are gone, just gone. We're working on things like autologous therapies, and one of the concerns using stem cell transplantation, and one of the things we're concerned about, what people are always saying, is that you can't possibly succeed at that because it's going to cost too much. So I would like to use the CAR T therapy as an example of how something that costs a great deal but actually cures the patient is valuable and should have lasting value even beyond the cost of drugs to treat that patient for the rest of their lives.
Hey, can I just jump in? Does that fact that 80% of those cell lines are not what they say they are, does that mean what I think it means? A lot of that research?
It does. It does mean exactly what you think it is. I've been doing a lot of research on this because I'm trying to get a company that does forensic analysis. I mean, they're actually doing they're one of those CSI type companies, right? They have a quick identification method. I'm trying to get them interested in the cell field so that they could be used as a method for quickly identifying your cell lines.
You know, it's too bad we don't have a break here because next door they're running a session of financing stem cell work. So,
should I just go over there right now?
Would you like me to make a statement? 15 years ago, this is to address the whole standardization question, which I think dovetails pretty well with your intent. 15 years ago, we founded Division 4, FO4 of the American Society for Testing and Materials, division on tissue engineered medical products, which has been extant ever since, and attempting to identify standards for tissue engineering or regenerative medicine products. It's now run by Tony Radcliffe. I would encourage all of you to look into it, primarily because that process allows for a coordinated stakeholder negotiation of what final standards really are, and allows for that funneling effect that you described earlier. And in the end, hopefully, we'll come to conclusions that we can live with and that can help guide better processes through the FDA. What surprised me the most in the past year about regenerative medicine was actually a form of retrenchment in an area that I thought was the most promising toehold for regenerative medicine. And that had to do with the utilization of stem cells from autologous tissues that have been manipulated in the operating room. This is a process that's been going on for quite some time, particularly with adipose stem cells, where adipose tissue is harvested, enzymatic digestion is done to be able to receive the stromal vascular fraction of the adipose tissue, which has been identified as being regenerative in many settings, certainly many nascent studies, but very promising studies. Recently, the FDA has begun to declare the functions of tissues in kind of odd ways. For example, as I mentioned yesterday, in a panel. The FDA has described adipose tissue merely as a structural tissue that provides cushioning for the body, and therefore the addition of adipose tissue to reconstruct a breast is no longer considered to be an appropriate homologous use. So there clearly seems to be the need for a lot of dialogue with the FDA now by all of us to begin to think about the fact that we're dealing with a very major metabolic organ that does far more than simply cushion the body. And we have to come to some terms with what we think these tissues are really doing and how much manipulation in an autologous setting really should be allowed for the care of patients, because if we don't get the toehold there, it's going to be very difficult to use heterologous tissues or allogeneic tissues at the stem cell level and to be able to use them for implantation as well.
Anybody have a response?
So I'm not opposed to using adipose tissue-derived cells for structural repair and things like, plastic surgery. What bothers me is that that particular type of cell has been touted all over the world, and unfortunately a lot in my city, San Diego, as being a miraculous cure for every disease. There's one company in San Diego that has claimed that adipose-derived cells can cure Parkinson's disease, and they'll just pull them out, clean them up, and inject them back into you, and you'll be better. So that kind of thing makes me completely crazy. And one of the things that makes me crazy about it is that the FDA has failed to enforce their own regulations. Whether they're right or not, there should be some kind of line drawn in the sand that says you can't trick patients into therapies that cost a lot of money and will do them no good. That's irresponsible. Yeah, you want to? Yeah, okay, I'm going to let him add something in a second. So it's been really frustrating to those of us who receive phone calls all the time asking whether this or that clinic is a good place to go for the disease we have. I don't speak out in, just to make it very clear, I do not mention the names of any clinics because all my friends who have mentioned them have gotten sued. So I'm in generalities, these clinics are not providing what they promise, and they are only selling disappointment, not cures. Now you can.
Yeah. So I'm in complete agreement with to being against hyping of these therapies. Recently, 2 weeks ago, we ran a meeting in Mallorca, Spain, called Cells Orthopedics that was designed specifically to identify what is allowable from a regulatory point of view in the various countries of the world, and how patient advocacy can begin to expand those regulations only on the basis of valid clinical data that's been generated. So we're going to need to have a stepwise approach following, we have to accept the regulations, but we also need to begin generating the data we can to expand utilization and certainly condemn any attempt to hype these therapies beyond what the regulations allow.
Yes, I also have been concerned with this shopping list of therapeutic with a single cell product. However, in the case of adipose-derived cell products, and I have a conflict because I'm the scientific advisory board of Lipogen, but I've been incredibly surprised because they came to our cell transplant center to say, can you help us explain why we observed these results in orthopedic application, in femoral head necrosis, in bone cartilage repair, besides the traditional plastic surgery. And I think we have a big role as academic institution and university to be the funnel and screen and try to understand from some of this clinical practice that sometimes are not done in randomized prospective fashion, but to say, wait a minute, there is something interesting. We see cartilage repair with thickness increasing from 0.7 to 1.5 millimeter. Let's look into this. Or we see femoral head necrosis repair by injecting adipose-derived tissues. To me, has been intriguing enough to begin saying, okay, let me help. At the same time, there is a very important deadline because April 13, there will be the meeting at FDA on this proposed guideline that to me is amazing. It doesn't make any sense to say that now breast reconstruction has been done post-cancer radiation injury for decades, and now to say you can no longer inject fat tissue because the breast is supposed to do milk and have ductal is like completely obscene as a as an attempt. But if we don't respond and if we don't make this regulation easy enough or reasonable enough to be able to do innovation trial and trying to understand the biology, the mechanism, and do validation, hands-on due diligence, we're an uphill battle. We cannot be treated as drugs.
Okay, I just wanted to make a comment that I think one thing that I find very encouraging is how, as a society, we're becoming much more mature in our approach to science and knowledge. And we are starting to become full partners with society as scientists. I think scientists are generally getting a little better at communicating with the public. But this issue to do with the FDA, I think, is an excellent example of where the advocacy groups and the informed public need to be picking up the phone, writing letters, and demanding that the FDA actually does its job. Now, if another scientist picks up the phone and says, hey, I want you to do something about a clinic, or I want you to do that, they're going to go, yeah, that's the scientists. They're really protecting their own interests. They could say that. And in some senses, they might be right. But if enough of Joe Public is picking up the phone and demanding action, that then translates into votes, into policy decisions.
So the first day of the meeting, we had a talk by the next FDA director, which I thought was really fascinating. Yeah, it was really good. And when I asked him what we should do about clinics that we think are offering therapies, that are offering treatments that actually are fooling the patients, is that we should contact the FDA. That is, that's what we get to do. The FDA may or may not investigate after that. But if you don't contact the FDA, that's not, they'll never notice. They'll never get to it. They're overworked, and it's, I understand that. It's frustrating, but I understand it. However, his advice was contact them. Tell them what this group is doing. Tell them that you don't think that it's legitimate use of these cells. So I plan on doing that, and I hope you plan on doing that too.
As the gods would have it, we have somebody from the FDA here.
Okay. Well, thank you so much. I'm so glad I'm here. Thank you for the excellent panel. My name is Xiaohong Fan. I'm a hematologist and oncologist. I work as a medical officer for SABR. FDA have a few center, but the SABR specifically regulated biologics. So my immediate office director, Dr. Wheaton, did give a talk at the beginning, and our big boss, the number one, Dr. Caliph, was there too. But anyway, I want to tell you a few things. First of all, I have to declare right now, is on top of my head, it's my personal opinion. Haven't gone through internal discussion, so I take responsibility for my comments, a personal opinion. I act here, but as official, it's like a scout. FDS scout. My bosses, they're gone, they're so busy. But they left me here. I actually want to hear from you, want to collect your concerns, your opinions, how to make us to do better work. I think we have the same goal. It's to facilitate, our goal is to facilitate for the safe and effective treatment for the patient. So we're patient-oriented. That's why I'm here. So I'm glad I hear your concerns. I will bring back, for example, the adipose tissue issue. I work for more gene therapy and cellular therapy oriented. So I'm not familiar with the tissue, that part. But I hear your concern. I will bring back to our specific division and say anything if they can help to address. Second, I would like to tell you is we do have actually the president of, as you said, there's a concern for some clinical, some practice is not in line with or violate some regulations. Personally, I had files, and because of, we call it the whistleblower, and then we do have the enforcement divisions. They start to inspection, and eventually we do have some enforcement. But I know it's very hard. It takes about 2 or 3 years, eventually disqualify the investigator or disqualify the data. It's very hard, but we have process. It's not easy, but we have the procedures, process in place. So I want to make sure of that. Third point, maybe I want to point out that like stem cell, like this area, fascinating and advanced very fast. As the nature of our job regulator, I think I repeat myself with, I realize that so too. We are behind. It's just nature of this profession, a little bit behind of the innovation and discovery. When you have issues, so that's why it's important to have this dialogue. I really welcome you all come, your questions or your proposals, talk to us. And I would like to tell the sponsors for any clinical trial you would like to introduce your product into clinics and come to talk to us. We have pre-IND meetings free. You don't have to wait for too long, get everything ready. No, you have your concept, you have your proposal, and you request a meeting. It's not as difficult. Website, where you're gonna go, there is address. So you can send it to request. Then we triage to the different divisions of people involved to talk to you. Even you have pre-IND, we even have pre-IND. So I want to welcome you, come to talk to us, and we hope we'll, in this new area to help move the field forward.
So I just wanted to say a word in response to your first point about people coming to you and having proper dialogue and telling you what we need you to do. Speaking as the representative of the gerontology community here, I just want to congratulate the FDA, first of all, on something that many of you may have heard about that happened quite recently, a few weeks ago, where the FDA moved forward to approve a clinical trial basically for intervention in aging. This has been a real bone of contention between the regulatory community and the gerontology community for many years, the essential feeling being, aging isn't a disease, therefore you can't have drugs for it, therefore you can't have drugs approved for it. And this was perceived as being a big chilling influence on the involvement of big pharma in, of the medical industry in general, in terms of development and translation of such interventions. I think that it's important to say that this is only the very first step. What essentially happened was that metformin administration was approved for a clinical trial, and the endpoints essentially are defined in terms of looking at multiple indications of the age-related ill health that people have been exploring. And I think this is the way to go, to define aging essentially as comorbidity, as multimorbidity. But we need to go a lot further, not only in terms of the endpoints that are defined for such trials, but also in terms of what the intervention actually is, because since aging is such a complicated and heterogeneous phenomenon, it's vital that we look much more favorably at combination therapies, at multiple things being done to the same people at the same time, as well as, of course, preventative therapies that are applied to people who are not yet demonstrably sick. These things, I feel, are going inevitably to be the future of medicine across the world as the ill health of old age so hugely dominates the medical profession and medical industry. And the only question is how soon. So I think the FDA and counterpart organizations across the world have an extraordinarily important leadership role to play in communicating with the gerontology and regenerative medicine communities to enable and facilitate the regulatory approval of combination preventative therapies for multiple simultaneous conditions.
Usually we go through it from my experience. For FDA is a data-driven agency. So we look at what you have. You give us the data, what's your rationale, what's your supportive information, then we evaluate. In terms of indication, we have to also look into the nuances, which population, what you have done, what you demonstrate. That's important to come to the endpoints. So how to measure it, how to compare what's available therapy there to give this convincing or persuasive data to show that it's really efficacious and also safe. So we needed the two. In the past, a long time ago, at the beginning, safety, but actually, both safety and efficacious. Some of the therapists maybe have less concern about the safety. So I give the patient that they are fine, right? But then you do also have the effectiveness, effectivity or efficacious results to demonstrate they have both sides there. That's one part. So we come to the details. What's the implication? We have to buy that data-driven evidence. If that clarify, I'll answer your question.
Yeah.
Thank you.
Someone patient over here.
Oh, my name is Arnaz Robin. I'm also from San Diego, Dr. Loring, and I've heard you over the years talk about medical tourism and rally against medical tourism, which I completely understand. But being from San Diego, I know there are stem cell clinics in Tijuana. And in popular culture, I follow sports. Bart Starr, who was a former Green Bay Packer quarterback, goes down there for treatments to Tijuana. Kobe Bryant goes to Germany for stem cell treatments. This is not something that's going to go away, and it raises the public's awareness of it, especially when it's popular figures in the media. What I wonder is there any way we can at least learn something from these human clinical trials going on? I know the FDA is going slowly, and I know we have to worry about regulation. And I know that some of these trials, you know, they say they're injecting MSCs, but who knows really what's in the mix. I understand all that. But there is a great wealth of knowledge that can be learned, at least from, if not collaborating, at least reviewing what is going on already. And this especially applies to Dr. Gray's work. I've been following your work for a while, and I'm not getting any younger. And even the idea of being able to I know, but I'm not getting any younger. And even the idea of being able to stop aging makes me want to try treatments on myself as a well-informed scientist. So I want to know, is there at least an opportunity to use their data, even if it's not always the proper protocol?
Yeah, I've actually thought about this and suggested this to some companies. If they have, if they want to charge patients for therapy, and they obviously are doing that, if they wanted to get approval eventually and show that their therapy is effective, they should contact the FDA and arrange to do this in the right way. One of the things they need to do is to follow up on the patients. And right now they don't do that. They just throw them out the door and say, well, if you don't feel well, come back and we'll give you another one. So they could do it. And they just, and I would love it if one company just decided to do that, because then the data wouldn't be lost. And there would be, then we'd know one way or the other. I'm not assuming that they're all useless. I'm not assuming that that they're all useful. But we just don't know, and that's because they don't follow through.
I'm not sure how many in the room know that there's a very interesting new angle that approaches what you've asked for, and that it's coming from the most unlikely quarter. The government of the nation of Nicaragua has approved the development of cell therapy centers within their military hospital system, and protocol-driven cell therapy with the characterization and follow-up that I think would appeal to all of us is in the process of being carried out. And it's it's those low-hanging fruit areas where I think we begin to learn things clinically that reassure all of us about moving forward, and hopefully that process will be carried out to the level of standards that we would all accept, because I think it's one of the rarest opportunities we have right now.
Do you think they will?
Yes, I do. I do think they will.
Are they in contact? Are they in contact with the FDA to find out what the standards should be?
I'm not familiar with all of the mechanics of their operation, but I do know that the will to do standard operating procedure-driven care is there, and that protocols will be followed that are reviewed by very appropriate people here in the United States, and that the process is underway, and it's probably amenable to the approaches that we want. But I just want to make you aware that this is something that's very new. It's really within months, and it's something we should keep an eye on.
I'm just going to state the obvious, that with these clinics, if you're thinking about using their data, we've already established that they're not approaching the enterprise in good faith. Why would you expect their data to truly represent the results of what they did?
Yeah, I'm delighted to hear the comments of the others to your question, because even though I've never spoken to any of these people about it, my attitude and my interaction with such companies has always been exactly as Jeanne first said, and so on. The question is, as you say, what to do about it. And the fact is, these companies do not have very much incentive even to think long-term about getting their stuff approved. They're making plenty of money as it is because these therapies are expensive and there are people out there who are willing to find the money to pay for them. So I've had these conversations and tried to persuade people to gather follow-up data, long-term follow-up especially, and it's completely ineffective. They've just not got the incentive. So I think we need to think much further outside of the box in terms of how to actually achieve the data gathering that you're talking about. The only thing I've thought of, and this is really outside of the box, is for the regulatory authorities or other government agencies or perhaps other non-government agencies to actually find funding to pay people who have been treated by whatever company in whatever jurisdiction to actually come in and subject themselves to follow-up, to actual, monitoring of their health in various ways, connected or not connected to the condition for which they got the therapy in the first place, so as to gather the data independently of the companies that are performing the therapies.
Good morning. My name is Gary Susser. I am a father of a 15-year-old who is quadriplegic, cerebral palsy, and cortically blind from an ischemic event at birth. In addition to that, I'm a consumer rights lawyer. We have been through the path as discussed because in 03 and 04 we went to California before Prop 71, went to Tijuana for stem cell therapies. We did it a few times, and since 03 and 04 the science has caught up. We've done a turnabout to see the hucksters where we live in South Florida doing full-page ads from Tampa to Miami to Boca Raton. And as a consumer rights lawyer, I invite the panel, and I'll give you my card, to help me use the Florida's Unfair and Deceptive Trade Practices Act to stop these clinics. And I can act a lot faster than the FDA. I could put these people down if I have the right people backing us and the right patients who've been scammed for tens of thousands of dollars, $50,000s of dollars, hundreds of thousands of dollars by these clinics who are promising false hope and false cures. My bona fides are such over the past 12 years, we actually appeared with Dr. Joanne Kurtzenberg on 60 Minutes when we helped bust a defrocked doctor selling stem cells over the internet. So we are in the fight. There are patient advocates. I, for one, will take this fight to the courts with the right people and the right clients. I've had unfortunate success, not success, because people don't want to go out in the public, be exposed with the attendant publicity that will be attracted. So if anyone in this room or on the panel has patients in Florida, whether it's Jacksonville, Tampa, Miami, or Boca Raton, who've been scammed by these hucksters and want to pursue this, you have an advocate.
So I have a question for you about that, because you being the legal expert can answer this perhaps better than any of us. What is your source of optimism that you would actually succeed in such a legal process? It seems to me that the difficulty is, these people who are supplying these treatments are innocent until proven guilty, and, it doesn't matter how regulated a treatment is, most treatments don't work every time. So, how are you going to prove that these people are really defrauding their customers?
The burden of proof, if you put 6 people on a jury, because it would be a jury question, what is unfair and deceptive? When you look at the advertising that's going on, and they can't escape their printed advertisements, and when they're giving you false promises and false hopes, and some of these people are board-certified orthopedics, and what they're doing is they're running a printing press for money, and some of them are hematologists. They're just doing things, and what they're advertising is what you hang them on. So when you put that up there and you bring the real science to the room, which is members of this panel and others in this room, and you sit there and you tell the public, this is what real science is, and what they're promising is not real, it is false, it is unfair, it is deceptive, you use that community standard. And that is a subjective standard, and the profiteers and the avarice and greed will come forward, and they will be addressed by a jury of their peers. And it's not medical malpractice. This is just consumer rights litigation, and that's the way to approach this. faster than the FDA can do it.
This exceeded far beyond my dreams. I wanted interaction. We've just got about 10 minutes to go. So next two questions, please make them quick.
Yeah, my name is Hector Manana. I am the head of the Oncology Research Unit in Mexico City, and I am also the founder and the current president of the Mexican Society for Stem Cell Research. And one of our goals during the last few years has been trying to fight those pseudo-clinics. We call them pseudo-clinics. There are lots in Tijuana, in Juarez, in Monterrey, and some other cities in Mexico. I'm not a physician, I'm a PhD. However, probably 3 or 4 calls every month I get from parents asking me if they should go to Tijuana because they have been offered a cure for his or her children or child with Down syndrome. I mean, Down syndrome. And they know nothing, and they've been offered a cure. So we are fighting very strong against those pseudo-clinics. The FDA equivalent, the Mexican FDA, it's called COFEPRIS. But I have to say, and I hope there are no microphones in here, but, well, except this one. But the Mexican FDA is not well prepared yet for regulating stem cell application. Legislation and regulation is still a work in progress. They are working on that, but they are not ready yet. There is no a particular document. So those pseudo clinics are taking advantage of that, and they are offering so many things, and they are just playing with the people's needs and hopes. And I don't know, there should be a way in which Americans and Mexicans work together trying to stop this, because it's a problem that is harming both countries, not just one. Well, actually, the world, the entire world.
As I said, we're running late, and Aubrey de Grey has another session in 10 minutes. What I'd like the panel to do, based on what you heard, based on what you didn't get to today, because we had like 16 questions, but I'm glad you guys got involved. Thank you very much. This is what we wanted. Why don't you do a wrap-up statement about what you've heard here, where you think it's going, or whatever.
Yeah, I think this is very great, everyone. Thank you so much for participating. But I think maybe the most important things that we've heard today are that the FDA is open to listening to us, that they're data-driven, but the data has got to come from well-codified sources. And so, Camilla, when you stood up and talked about that, you know, if there are risky things to do, let's do them in environments like yours and other investigators here who really know how to do these things properly, so that when the data comes out the back end, we can rely upon it.
I'm really interested in finding ways to prosecute people who mislead the public with stem cell clinics. And I have, in fact, helped with some cases. I have to, as I said earlier, I have to be anonymous because they do sue people. But I'm really proud of the fact that I've caused one California doctor to lose his license. Not that I think that'll make much difference to him. And I also helped the FBI with the famous case that was shown on 60 Minutes. I think ultimately 60 Minutes is probably the quickest way to go, if we could just get them to do a whole bunch of these. Investigative journalism is probably the most effective way to draw people's attention to this. I'd be interested in finding out what laws there are in other states that might be used to have some kind of impact on these clinics.
I might add, after what Jeanne said at the beginning, I think she'd also like to prosecute all the poachers who are basically causing a lot of species to go extinct, correct? Okay.
No, I mean, it's like Al Capone on tax evasion. Whatever the law you can find to pursue it is the best. I really want to congratulate, actually, the whole conference. It's such a nice conference to come to. It really is truly collaborative. Everybody gets on well. Actually, it's sometimes a little disappointing how nicely everybody plays together at this conference. It certainly doesn't happen at every conference I go to. So I basically want to repeat just how important it is that we talk to you and you talk to us. I think sometimes people feel editors are a little bit removed and don't look behind the curtain. It's just not the case. We need to hear from you as well as expect that you trust us to pursue the goals that we all have for regenerative medicine.
Yeah, I certainly echo everything that the others have said. I think really, since we're coming near to the end, sort of the last day of the conference, it really, the intensity with which these topics have been discussed in the past hour reinforces the very wise decision that Bernie and his colleagues have made over the years to have an entire track on policy and ethics and society in this conference. It really shows how engaged the scientists and industrialists who come here are in those other issues. And speaking, of course, as the rest of us as editors of Liebert journals, I want to echo what Graham just said, that it's vital that we should be given the wherewithal to understand what issues we need to be focusing on, what contribution we can make to improve the quality of science that is published, as well as, of course, the quality of medicines that are eventually made available to the public.
So I want to thank the panel. As you can see, they're very knowledgeable and very opinionated. I really want to thank you all for getting involved. I'm going to see next year if we can push this to maybe 2 hours, because a lot of things we didn't get to. Maybe we can even bring some cheese and wine to get the discussion going even more. And that's not saying this is not serious business. I was just really glad you all got involved, and there's a lot of things we didn't get to maybe next year. Thank you so much, and have a great rest of the conference.